I would like to thank very much the Hellenic Urological Association and the European Association of Urology for giving to me the opportunity today to share with you our thoughts on the post-fertilization effects of primary testicular damage. Which is the mission of the spermatozoa? Does sperm quality affect the oocyte fertilization process? Does sperm quality affect early embryonic development? Does sperm quality affect embryonic potential for implantation? Which is the mission of the male in the earth? Previous research efforts in our laboratory have indicated that embryos generated from the fertilization of oocytes with spermatozoa recovered from rats with primary testicular damage due to chronic renal failure have lower potential to further develop in vitro and subsequently implant into the uterus. This is the varicocele model in the rat. In photograph 1, white arrows indicate the control normal left testicular vein, whereas the varicocele model appears in photograph 2, where the white arrows indicate the dilated left testicular vein, confirming the development of varicocele. Embryos generated from the fertilization of oocytes with spermatozoa recovered from rats with testicular damage due to experimental varicocele induction have lower potential to further develop in vitro and implant subsequently into the uterus. The blue arrow indicates an oocyte fertilized with spermatozoon received from a varicocelized rat. The embryonic development was arrested at the morula stage. The blastomeres demonstrate variation in their size. Blastomere size is non-equal among the cells. This clearly indicates low embryonic quality. The white arrow indicates another oocyte fertilized with spermatozoa from a varicocelized rat. The development of this embryo was arrested at the morula stage, white arrow. Blastomeres demonstrate variation in their size. The size of the blastomeres are not equal among the cells. This is an indication to confirm that that embryo has low quality. Recent studies in our laboratory have demonstrated that embryos generated from the fertilization of oocytes with spermatozoa recovered from rats with testicular damage due to experimental cryptorchidism induction have lower potential to implant into the uterus. These results are very exciting because the newborns delivered by females that mated with crypto male animals had significantly lower body weight compared with the control group. That study demonstrated that unilateral cryptorchidism induced testicular damage can significantly affect the contralateral testis, as well having further deleterious post-fertilization effect on the development of newborn. And the most exciting finding was that treatment of the father animal with antioxidants could partially improve the testicular damage bilaterally in the next generation, in the newborns. Other studies in our laboratory have indicated that human or rabbit oocytes fertilized with early chromosomally haploid male gametes, either with secondary spermatocytes or with round spermatids, demonstrate lower potential for implantation. The preliminary conclusions from the initial research efforts indicate that there is a paternal influence on the early embryonic development and capacity for implantation. The mechanisms mediating the above effects post-fertilization remain to be elucidated. The mission of the spermatozoon clearly is not only to fertilize the oocyte. The sperm transfers genetic and epigenetic as well factors important for the early embryonic development and the subsequent embryonic capacity for implantation. Therefore, it appears that there may be a functional defect in one or more ooplasmic contributions of immature male gametes due to the immaturity of the cell, or of the mature male gametes recovered from subjects with primary testicular damage. The contributions of the male gamete to zygote can be classified into genetic contributions, such as the nucleus DNA, and into epigenetic factors, the reproducing element of the centrosome. We are aware that the female centrosomic material is dormant, is inactive in the oocyte, and nature in the human being has given a priority to the male centrosomic material. So the centrosome in the human being is paternally inherited. Phospholipase zeta is a very important contribution of the sperm to the zygote because it is the reason of the main factor for oocyte activation, development of the female pronucleus, extrusion of the second polar body. Histones represent important as well contributions of the male gamete to zygote, having an epigenetic important role in the first hours post-fertilization. The major objective of this current communication is to understand better the alterations in the zygotic development that may be attributable to defects in sperm genetic and epigenetic factors, probably due to the origin of the spermatozoa from a malfunctioning testis. A recent research target is to investigate whether the pattern of blastocyst hatching depends on the quality of the spermatozoa that had fertilized the respective material. The blue arrows indicate the point of the exit of the embryonic mass through the zona pellucida into the uterus. This hatching process appears to be dependent by the male gamete. So it is the sperm quality that regulates the blastocyst hatching pattern, and the sperm quality is responsible for the presence of an outward pattern of blastocyst hatching or an inward pattern of blastocyst hatching. And the outward pattern of blastocyst hatching includes a large space in the zona pellucida for the exit of the embryonic mass through the zona pellucida. Inward pattern refers to a narrow space in the opening of zona pellucida for the exit of the embryonic mass through the zona pellucida towards the uterine cavity. But because there is a narrow space in the opening of zona pellucida, an embryonic stress may be caused, and the overall result may be lower embryonic implantation rate, exactly due to the mechanical embryonic stress. Human blastocyst appears here, developed from the fertilization of an oocyte with a spermatozoon recovered from a man with varicocele. When this blastocyst is cultured for some more hours, it appears that the hatching process demonstrates the inward pattern. The blue arrows indicate a narrow opening in the zona pellucida. The embryonic mass exits towards the uterine cavity, but this narrow exit opening may cause a mechanical stress in the embryonic mass, resulting in lower implantation rate. The opposite is the outward pattern. Human blastocyst develops in this image from the fertilization of an oocyte with a spermatozoon recovered from a man after varicocelectomy. Initiation of natural hatching process is indicated. The blue arrows indicate the outward pattern. The blue arrows clearly demonstrate that the opening in the zona pellucida is large. The embryo comes out into the uterine cavity in a very smooth manner, and therefore mechanical stress on the embryonic mass is avoided. These blastocysts have higher potential for implantation and induction of pregnancy. DNA integrity, a reproducing element of centrosome, phospholipase zeta and histones represent the contributions of spermatozoa recovered from healthy subjects. However, the above described research efforts tend to suggest that there may be a functional defect on DNA integrity, centrosomic material, phospholipase zeta, histones mechanisms in subjects with primary testicular damage. And these functional defects may represent the connective link between primary testicular damage and lower embryonic implantation rate after the fertilization of oocytes by male gametes recovered from humans or animals with primary testicular damage. A powerful technology is available in several laboratories. Real-time lapse cinematography. This technique provides high-resolution, continuous imaging in which the various microcomponents of the cells can be clearly distinguished under high magnification and monitored in a real-time fashion. This equipment can be observed in this image. Culture conditions for the oocyte in the class cube, you may observe, involve temperature of 37 degrees, pH of the culture medium 7.37, exposure time for digital images is 50 milliseconds, and there is an auto-focusing system. In these initial efforts, Group A included healthy normozoospermic men. Female factor had been confirmed. The female partners were characterized by fallopian tube obstruction. Group B included infertile men with varicoceles who did not undergo varicocelectomy. And Group C included infertile males with varicoceles who underwent varicocelectomy. In the last group, spermatozoa were collected as the respective publications demonstrate 5 to 13 months post-varicocelectomy. The inward hatching pattern indicated by red letters demonstrate that the values were significantly higher in the varicocele group, 86%, compared with the varicocelectomy group. It means inward hatching pattern characterizes the animals or the humans in this study with primary testicular damage due to varicocele. The outward hatching pattern in men after varicocelectomy was 71%, statistically significantly higher compared with men who underwent assisted reproductive technology without pregnancy in the beginning and then nothing else. This was the simple varicocele group, 14%. So varicocelectomy had a beneficial effect on the blastocyst pattern of hatching, and maybe this is the reason of the beneficial effects of varicocelectomy on the implantation rate. The above initial studies and the above publications allow us to state vividly that the primary testicular damage results in defects in sperm genetic or epigenetic factors. The next result is that most of the blastocysts demonstrate inward hatching process. And the next result is the presence of embryonic stress during blastocyst exit through the zona pellucida, which is the overall result, lower implantation rate due to the mechanical stress on the blastocysts that are released through the exit in the zona pellucida. However, these techniques allowed us to study additional events in fertilization process. IVF technique, blue arrow indicates a sperm penetrating the zona pellucida. After a few hours, female and male pronuclei can be observed by the arrows. This is the evidence of fertilization. Oocyte cleavage some hours later, and development up to the 4th cell stage by the fertilization of an oocyte by a healthy sperm. However, if we see the dynamics of embryonic development after fertilization of an oocyte with a spermatozoon in ICSI techniques recovered from a man with varicocele who did not undergo varicocelectomy, we can see the first and second polar body with the blue and green arrows, and the female pronucleus indicated by the red arrow, and the male pronucleus indicated by the purple arrow. The oocyte, after some hours, started the cleavage process, and the cleavage point is indicated by a blue arrow, but cleavage cannot be completed, and the oocyte resumes its original shape. Cleavage cannot be achieved. And after some hours later, cleavage can be obtained by a second effort of the fertilized oocyte, and we have two-cell stage development. It appears embryonic dynamics are influenced when the sperm is retrieved from a man with a primary testicular damage. And actually, if we culture these oocytes that have been fertilized with spermatozoa from males with primary testicular damage up to the blastocyst stage, inward pattern may be very often seen as indicated by the narrow opening in the zona pellucida confirmed by the blue arrows. This is the inward pattern when the oocytes are fertilized by spermatozoa from animals or humans with primary testicular damage. On the other hand, fertilization of oocytes with healthy sperm or spermatozoa from a man after varicocelectomy show a smooth exit of the blastocyst through the opening of the zona pellucida towards the uterus without the occurrence of mechanical stress on the embryonic mass. This technology allows us to see also some other differences in the fertilization process. We have been educated in medical schools that only one sperm comes inside the zona pellucida. However, the blue arrow indicates a leading sperm and the green arrow a following sperm. Always two spermatozoa penetrate the zona pellucida. And when the leading sperm comes into the cytoplasm, the following sperm stops the penetration through the zona pellucida. And after some hours, the leading sperm is inside the oocyte, and this is confirmed by the presence of male and female pronuclei indicated by blue arrow and green arrow, and the following sperm has stopped his entrance into the oocyte. Definitely, the male is something more than a missile, and the male represents a multifunctional instrument, and we need technology that allows us to take more messages from the fertilization process, because in males with varicocele, sometimes both spermatozoa come inside the main sperm and the following sperm into the cytoplasm, and polyploidy is not rare in ICSI techniques when spermatozoa from males with varicoceles are used. Philosophically speaking, these preliminary results may allow us to state clearly that pathophysiologies of a male organ like varicocele affect physiological events within the female body, like the blastocyst hatching in the uterus. This might be consistent with the Holy Bible words stating that the first female human being, Eva, was created by a male organ, the rib of Adam. And the blue arrow indicates within the uterus of Eva cytological events referring to the hatching pattern of the blastocyst regulated by the testes of Adam. So Adam may claim that he is the boss. Thank you very much for your attention.